Hello everyone, this video is about the nerve system of insects. In insects, like other metazoa, the nerve system is a major seat of coordination, learning, memory, intelligence, and behavior. The cellular units of the nerve system are the neurons. They are functional elements of the nerve system, constituting the ganglia, nerve cord, commissures, and nerves, and communicating a wide variety of receptors and effector organs. The neurons. These are derived from the embryonic ectoderm. Each neuron consists of a prominent nucleated cell body or soma and a long cytoplasmic filamentous extension of axon. The cell body of one neuron communicates through the transitional gap synapse with the axonal terminus of the other neuron in a linear fashion so that a continuous chain is formed along which the electrical disturbances or impulses can be transmitted from one part of the body to the other. Soma is provided with the fine processes called the dendrites, while the terminal end of the axon bears a group of fine fibrils called terminal arborization. This axon frequently gives off one or more short side branches, while the collaterals, which terminate into the terminal arborization. Multipolar neurons are mostly confined to the hypocercal. On the basis of functions, the neurons are often classified into three types: the sensory neuron, the motor neuron, and the association neuron. Sensory neurons, they are also called effector neurons and perform perception of the stimuli. They are bipolar neurons with a dendrite terminating into a sense organ or receptor and an axon running towards the central nervous system, and therefore their cell body is always situated outside the ganglia. They conduct impulses from a receptor to the central nervous system. So monopolar axon, dendrite, perikaryon or soma or cell body, terminal arborization, bipolar, multipolar. Sensory association motor neurons and target organ muscle. First, the sensory neuron. They are also called the efferent neurons and perform perception of the stimuli. They are bipolar neurons with a dendrite terminating into a sense organ or receptor and an axon running towards the central nervous system. They are also called the effector neurons and perform perception of the stimuli. They are bipolar neurons with a dendrite terminating into a sense organ or receptor and an axon running towards the central nervous system. Then the motor neurons. They are the efferent neurons. The soma of these neurons are situated in the ganglia and are monopolar, devoid of dendrites. Their axons form the motor nerve and innervate various organs of the body, while collaterals merge into the neuropil. They conduct impulses from the central nervous system to the effector organs. And the association neurons make association between the efferent and efferent neurons and are called the interneurons, internuntial or interpolated neurons. They are situated within the ganglia. They bear comparatively small cell bodies, mostly occupied by the nuclei, and are called the globuli. The giant neurons are running the total length of the nerve cord and represent themselves as the association neurons. Similarly, the transverse commissures are formed by these neurons. The neurons constitute a definite nervous system, and in insects, both the central nervous system as well as the sympathetic system are well developed. Besides these major systems, some bipolar neurons are distributed in the peripheral region of the body, innervating the integumentary hair. Similarly, some multipolar neurons form a complex nerve network beneath the integument, above the muscles, and on the surface of the gut. All these neurons constitute the peripheral nerve system. The next major topic is the central nervous system. First, let's learn a basic plan of central nervous system of an insect. It consists of a anterior dorsal brain and ventral double nerve cord bearing the segmental ganglia. Each embryonic segment primarily bears a pair of ganglia which are connected longitudinally by intersegmental connectives and transversely with or without the intersegmental commissure. The brain is connected with the first ganglion of the ventral nerve cord, the subesophageal ganglia by paired circumesophageal connectives. The head or cephalic region possesses two ganglia, the brain and the subesophageal ganglia. The brain is composed of three paired segmental ganglia, the preantennal, antennal, and intercalary ganglia, while the subesophageal ganglion is formed due to fusion of three paired gnathal ganglia, the mandibular, maxillary, and labial ganglia. Brain, frontal ganglion, corpus caadiacum, esophageal ganglion, corpus alatum, hypocerebral ganglion, proventricular ganglion, spiracles, transverse nerve, perisympathetic organ, median nerve. These are the anatomical portions of the insect nerve system. The thoracic region of insect is composed of three segments: prothorax, mesothorax, and metathorax, bearing the prothoracic, mesothoracic, and metathoracic ganglia respectively. The abdominal region of insect is basically composed of 11 to 12 segments. In primitive insects, the segments 1 to 7 possess a ganglion per segment, and it is found the abdominal region of insect is basically composed of 11 or 12 segments. The segmental ganglia supply nerves to the appendages, muscles, and organs, and a part of heart lying within their respective segments. Next, condensation. An extensive condensation of ganglia of ventral nerve cord can be seen in higher groups of insects. In primitive and lower groups of insects, all three thoracic and 8 abdominal ganglia are distinct and separated from each other. In higher insects, first abdominal ganglion often focuses with the metathoracic ganglion, or one or two abdominal ganglia may fuse with one another, and the total number of abdominal ganglia therefore reduces variability. The abdominal ganglia occur in maximum number 8 in Dictyoptera, while in other insects the number reduces to 7 in Orthitrum, Periplaneta 6, and 5 in Tabanus, 4 in Haematopota, then 2 in Syrphis, and 1 in Xenopus. High degree of condensation shows the presence of only one ganglionic mass behind the brain in Hydrometra, two ganglia, one subesophageal and the other composite thoracico-abdominal ganglionic mass in Musca and Calliphora. T ganglia, first subesophageal, second composite thoracic ganglionic mass, and third the abdominal ganglionic mass in Myopa. The ganglia, though differ in shape, size, location, and division of labor, exhibit common basic structure. The ganglion consists of the outer ganglionic sheath, the peripheral cortex, and the central glial mass or medulla. The brain is the most developed ganglia and functions as a master organ of association, coordination, other ganglia maintain single segmental coordination. Neurophile, play of perilurium, neural lamella. These are the histological parts of a ventral region for ganglia, ventral ganglia, along with the connective or nerve. Nerve root, giant fiber, neural lamella, glial cell nuclei, trachea, cell bodies. The next, the ventral nerve cord. It contains axonal processes of variable diameter and they are invested by the glial sheath. The glial sheath does not bear the lacunar or vascular system. The majority of the axons are uniformly smaller, measuring about 5 μm in diameter, but there are some much longer axons of about 60 μm in diameter and are called the giant fibers. The giant fibers are reported in the ventral ganglia of Odonata, Dictyoptera, Orthoptera, and some other orders. They are interneurons which run for a long distance without synapse. All axons of the ventral nerve cord are externally covered by a neural sheath. The neural sheath is composed of two layers, outer neural lamella and inner perineurium. The perineurium of the ventral nerve cord is often nucleated rather than cellular and also much reduced in the thickness. The peripheral nerves arise from the ganglia and innervate various segmental structures. The nerves may be of three types: motor, sensory, or mixed type according to the nature of nerve fibers they contain. The nerves are composed of axonal bundles enveloped externally by the nerve sheath. The nerve sheath is composed of an acellular basal lamina resembling the ventral neural lamella. It is followed by the layer of glial cells comparable to the perineurium. The glial cells are provided with the separate desmosomes, the gap junctions and tight junctions. At the center, the bundles of axons are tightly packed. In small nerves, however, a layer of glial cells is wanted. The axons contain the exoplasm with the mitochondria, as only the cytoplasmic organelles and transport secretory droplets from the cell body to the terminal end. Then the neuromuscular junctions. The motor nerves may contain three types of fibers: the fast, slow, or inhibitory, and make intimate contact with each muscle fiber. The nerve divides into fine branches which pass longitudinally over the surface of the muscle or may be invaginated into the muscle fiber. The terminal end of the axon divided the surface of the muscle and axonal fine branches form a sort of claw-like structure. A single muscle fiber may be innervated by a single or many axons. The site of neurotransmitter contact is called the neurotransmitter junction or the neurotransmitter synaptic contact. It may be designated as the fast, slow, or inhibitory in accordance with the type of nerve fiber. The glial cells are absent along the nerve-muscle interface. The plasma membrane of both axon and muscle fibers come close to each, leaving a gap of 100 angstrom. The axonal end contains about 1,000 synaptic vesicles of 250 to 450 angstrom diameter loaded with the neurotransmitters L-glutamate and acetylcholine or gamma amino butyric acid inhibitory fibers. The next is about the brain. First, let's have an introduction about the gross anatomy. The insect brain represents the supraesophageal ganglion, and it lies above the esophagus in the oculofrontal region of the head. The insect brain represents the supraesophageal ganglion, and it lies above the esophagus in the oculofrontal region of the head. It is a bilobed structure. Both the lobes are well differentiated from each other by a distinct median furrow that is externally covered with air sacs and fat body. The aorta runs ventrally and opens in front of the brain. The insect brain is divided into three dorsoventral regions: the forebrain or protocerebrum, and midbrain or deuterocerebrum, and the hindbrain or pectocerebrum. The protocerebrum is the anterior dorsal large region of the brain representing the fused paired preantennal or archicerebral ganglia. It bears on each lateral side The optic lobes receives the optic nerves. Nerves optic is from the optic lobes and ocellular nerve ocellary from the medium and lateral oscilla and it gives out the cardiac nerve. The nerve or porous cardiac innervation, corpora cardiaca and the aorta. In the diptocerebral, this is the middle part of the brain is in the anterior ganglia. It gives out a pair of lateral lateral nerves innervating the anti-pulmonary and the nerve. The diptocerebral is surrounded by soon divided into two branches. The lateral nerve innervating the labrum and the frontal connective terminating into the frontal region. The ventral part of the brain representing the ganglion of the intercalary segment. It gives out a pair of labrofrontal nerves. Okay. Labrofrontal nerves. The nervi labrofrontalis, which soon divides into two branches. The labral nerve innervating the labrum and the frontal connective. The trictocerebral lobes are connected with the subesophageal ganglion by the circumesophageal connective. Then microanatomy. The brain sheath, cerebral neurolemma. It is composed of two layers, the outer neural lamella and the inner perineurium. The neural lamella is non-cellular, while the perineurium is a cellular layer. The neural lamella is a thick layer composed of the protein, collagen-like fibers having repeated banding patterns. So when we try to learn about the basic structure of insect brain, there are different parts like first intercerebralis region, ocellular nerve, then medulla region, inner medulla, outer chiasma, lamina, anaris, medulla, antennar lobe, trictocerebrum, then circumesophageal connective, post-esophageal commissures, mushroom bodies. Okay, calyx, pedunculum, etc. Microanatomy. The brain sheath, the neural lamella is a thick layer consisting of the protein and collagen-like fibers having repeated banding pattern. The fibers are 130 angstrom in diameter and are randomly oriented through often arranged parallel to each other. The fibers are embedded in the matrix of amorphous granular substance containing mostly the mucopolysaccharides. The mucopolysaccharides show the chemical properties of chondroitin, dermatin sulfates, and keratin sulfates. The neural lamella is 150 to 200 angstrom thick in Coleoptera and 500 to 600 angstrom in Orthoptera, and in most of the insect it is differentiated into the outer homogeneous dense and inner less dense heterogeneous sublayers. It is traversed by a large number of tracheae. When we try to learn about the histology of the brain, different layers are a glial fold, lacuna, glial cells, axon, neurosecretory cells, nerve root, axons, trophospongia, neuron, neural lamella, outer layer, neurolemma, perineurium. Okay. The neural lamella is secreted by the perineurial cells after modifying into the fibroblasts and secreting the collagen fibrils. It provides mechanical support and protection to the ganglion as well as nerve cords in general and to the brain body in particular. The perineurium is a thick cellular layer lying beneath the neural lamella as its inner layer. It is composed of two types of cells. The cells of one type are rich in mitochondria, fat droplets, and glycogen granules. The adjacent cells are communicated with each other by a series of layers of cone-shaped tubes. These cells are deep-deleted the adipose cells contain cells which are often filled with an amorphous matrix embedding the collagen fibrils and producing the neural lamella. The perineurium is trophic in its function and this also acts as a blood-brain barrier. The cortex, it is the peripheral region of the brain and other ganglia. It is filled with the monopolar motor neuron and the glial cell. Some of the neurons are modified into the neurosecretory cells and terminate into the neurohemal organs. The neurons are classified into two periplaneta to 5 in California types according to the variation of the size in their soma and cytological features. Their cytoplasmic organelles include the mitochondria and Golgi complex predominantly, along with the microtubules and dense vesicles containing enzymes such as thiamine pyrophosphate and phosphate, adenosine triphosphate, and neurotransmitters. The neuronal bodies are closely surrounded by the glial cells with a gap of about 100 to 200 angstrom occurring between their plasma membranes. The glial cells often send some invaginations into the peripheral cytoplasm of the prokarya of the nerve cells called trophospongia or the canals of Holmgren. The glial cells occur in insects in the truncated condition but never an myelinated form as occur in the vertebrates. They invest on sheath and ensure entire body of the neurons and provide insulating and protective covering around them. They extend the process within the medulla of the ganglia neuropile to invest the axons. Around the axon, the glial sheath may be comprised of a single or many folds as layers. Sometimes a single glial sheath may enclose various axons, or a single axon may be encircled by a large number of glial sheaths. The glial sheath is also termed as a neuroglia, lemnoblast cells, or Schwann cells, and are classified into 5 or 3 to 4 categories in Musca and Formica. The giant axons are ensheathed by 7 to 8 glial cells folds spiraled around their periphery and called the mesaxon. The gaps occurring in the glial folds may be distended at intervals which are filled with hyaluronic acid and may serve as the nutrient and cation reservoir. The adjacent glial cells are connected with one another by the gap junctions, accidental junctions, or zonula occludens, or tight junctions, continuous junctions, or zonula continua, and desmosomes. These junctions simply contain the structural integrity of the system. The pars intercerebralis. It is the well-differentiated median and anterior dorsal cortical region of the procerebrum of the brain. It is bounded laterally by the corpora pedungulata, apically by the neurolemma, and ventrally by the central body. It is divided into two identical halves by a median furrow and specialized by possessing a distinct group of neurosecretory cells on either side of the furrow. These cells are termed as the median neurosecretory cells. Their axons from the axonal tracts which cross over each other at the middle of the brain and finally emerge out of the brain as the nervi corporis cardiaci interni, innervating the corpora cardiaca. Besides the medial neurosecretory cells, there are large number of neurons invested by the glial cells, and some of them are receiving the trophospongia. The lateral cortical region of the protocerebrum is filled with a large number of neurons and some neurosecretory cells. The large number of neurosecretory cells cerebrum is exclusively occupied by the neurons of the glial cells. The tritocerebral cortical region, moreover, contains a paired group of the neurosecretory cells, the ventral neurosecretory cells, which extend their axons and constitute the NCC3, or simply may fuse with NCC1 or 2. The next medulla. It is compactly filled with the neurophile. It is composed of a mass of axons of varying diameters. Their terminal arborizations investing glia and tracheoles. The axon fibers are at some distance arranged into the bundles of tracts which may be branched or unbranched. The glial sheath is reduced or entirely lacking in some area, providing a site for synapses among adjacent axons. The presynaptic membrane bears so-called presynaptic knobs of 1.2 to 2.5 mm diameter, each bearing a cluster of synaptic vesicles. The synaptic vesicles are of two types, small to large in diameter, containing various neurotransmitters. The protocerebral neurophile contains some distinct neurophile masses, the pons cerebralis or protocerebral bridge. The corpora pedunculata or mushroom body is the corpus centrale or central body, the corpora ventralia or accessory lobes, and the corpora optica or the optic lobes. Then the pons cerebralis, it is a medial neuropillar body. It is transversely situated in the frontal region of the protocerebrum as a lattice neurophile behind the pars intercerebralis. The globuli cells are lying laterally. It is connected with various neuropillar masses except the corpora pedunculata. The corpora pedunculata, there are two mushroom-shaped bodies, each on either side of the pars intercerebralis. Each corpus pedungulatum is separated into two parts, an anterior flattened cap or calyx and the posterior stalk or peduncle trending downwards ventrally. The stalk is distally divided into two branches or roots, alpha and beta lobes. The alpha lobe is also called the other stack, posterior root or tubercle exterior. The beta lobe is called the median root, inner stalk of the tubercle interni, and it is extended towards the midline in front of the central body. The corpora pedunculata are large and greatly developed in social insects, moderately in orthopteroid and coleopteran insects, and much reduced in Collembola, Odonata, Heteroptera, and Diptera. Each calyx bears large number of unipolar nerve cells, the globuli or association cells. Their axons run to the alpha and beta lobes along their peduncle and are restricted there only. The calyces and alpha lobes receive sensory fibers, while the beta lobes send or synapse with the motor or promoter fibers. The promoter fibers are the and corpora centrale. It is a median mass of neurophile at the center of the protocerebrum. It is an association center from which the premotor outflow from the brain to the ventral nerve cord is conducted. It is connected to various parts of the brain by a large number of axonal fibers with the pars intercerebralis, cerebral lobe, endocellular masses, and that consists of their globuli surrounds the two terminals of the neural cells of the pars intercerebralis. In some insects, it is divided into two structures, the ellipsoid body and the fan-shaped body. The ellipsoid body is a ring-shaped neurophile mass connected with the median and superior parts of the protocerebrum by the oblique ellipsoid tract. The tips of the ellipsoid body it is connected with the medullae of the ellipsoid body is a nucleophile mass which is composed of fibers arranging radially similar to the staves of a fan. Each stave is made up of two sets of 8 bundles, each consisting of 4 fibers in Schistocerca, and in Musca domestica, each stave bears 14 bundles of fibers, dividing into two sets of 7 each. These fibers are originated from each other by the protocerebral bridge and then into fan-shaped body. In some insects like Coleoptera, the central body wears a pair of small spherical neurophile bodies called the fibers running from the body arborate here where the endings of the fibers are arrived from other parts of the protocerebrum. Then the corpora ventralia, a pair of ventral bodies or accessory lobes lie in the ventrolateral region of the protocerebrum, and both the ventral bodies are connected with each other by a protocerebral transverse commission. Each ventral body is divided into two halves, an oval and an ocular. Each ventral body is connected with the dorsal protocerebrum via median fold and the ellipsoid body or ellipsoidal fiber. Some axonal fibers form parts intercerebral, also terminate into the ventral body. Then corpora opaca lobes and do not represent the ganglia. Each optic lobe contains a group of three neophile muscles. The outer lamina ganglionaris or lamina, the middle medulla externa or medulla, and the inner medulla interna or lobula. They are connected to each other. The lamina lies beneath the comacula axon of the reticular cells through the basement membrane. These axons synapse with the monopolar axons into the lamina, which run into the medulla after crossing over each other and thus forming the external or the first chiasm between the lamina and medulla. Similarly, the fibers running from the medulla to the lobule cross each other and form internal or second chiasma in between the medulla and the lobula. Around the lamina, the cortical region is filled with the glial cells, retinal fibers, a layer of unipolar neurons, and that of small multipolar neurons. The trachea are richly supplied. The medulla is composed of several layers of dendritic endings, cell bodies, and peripheral neurons. Some axons end into the medulla itself, while others run toward the lobula. The neurons or medulla are classified up to 15 times. The lobula is a single neurophile mass except the diptera and lepidoptera, in which it is divided into two structures, one cup-shaped and the other plate-like bodies. Some axons may run directly from the lamina to the protocerebrum, where the axons pass through the ventral nerve cord along either ipsilateral or contralateral or both the circumesophageal connector. The medulla is connected to the callus and the corpora pedunculata by the dorsal tract. The optical tracts are the anterior optic tracts running from the lobula to the optic tubercle in anterior region of the brain, and superior medial optic tracts running from the lobules to the alpha beta lobes of corpora pedunculata, and the inferior medial optic tracts running from the lobula to the protocerebrum, and transverse optic tracts, one on most of the insects. Two spherical ellipsoid masses, each situated in each lateral region of protocerebrum, and is called antennal lobes. The antennal lobe is divided into two halves, the dorsal sensory and the ventral motor. Both are anterior lobes ventral nerve. Each is a mixed type of nerve containing a group of sensory and that of motor fibers. Both the lobes are connected with each other by the deuterocerebral transverse commissural tract. Each lobe is connected with the ipsilateral corpora pedunculata by motor and sensory fibers, and also connected with the tritocerebral lobes. The tritocerebral neurophile bodies. Each lateral tritocerebral neurophile body or lobe is composed of mixed fibers, the motor and the sensory. Both the lobes are connected with each other by the tritocerebral transverse commissural tract. It runs internally or in some insects externally. It is site of mixed nerves, the labrofrontal nerves. Cerebral integration. The brain mainly functions as an association center which controls reflex responses in accordance with stimuli received from the sense organs of the head. It exerts complex forms of behavior by stimulating or by inhibiting the reflexes. The brain controls the excitability of motors. It regulates oviposition, stinging reflexes, and walking coordination by inhibition. The corpora pedunculata play an important role in regulation of complex behavior by acting as a switch and controlling the temporal and spatial pattern of the behavior. They are provided with an inhibitory systems for general activity. The mushroom bodies determine initiation type and response of singing in the orthopteroid insects. In the sound produced by orthopteroid insects, crickets and grasshoppers, the corpora pedunculata integrate the input from several receptors and channel organisms from the ventral nerve cord, which provides signals such information, the singing is initiated. Stimulation of the calyx inhibits recalling of other songs depending upon need. The central body, the singing instructions of the mushroom bodies are analyzed and transmitted to the effector neurons of the mesothoracic ganglia. The central bodies are, as well as the protocerebral bridge, are concerned with the control of respiratory movements. In the brain, particularly the mushroom bodies, are involved in the mechanism of learning of various types: habituation, conditioning, trial and error learning, association learning, latent learning, insight learning, and social learning. The brain maintains equilibrium of the body.